So, as you heard, my talk will be in English. It's really a big pleasure. It's an honor for me to be with you at your pediatric conference and to present you our new hexavalent vaccine. And we just presented it last week for the first time at a big international conference. It was the World Society of Pediatric Infectious Diseases, which took place in South Africa, in Cape Town. So I came from Cape Town to Cyprus, which was a long trip. But the good thing is that the weather is the same there and here. So it's really great to be with you on your beautiful island and on your very interesting congress. Before speaking on the vaccine itself, let me say a few words about the challenge of combination vaccines. It's a major area of vaccine research. And even if the antigens are already known, when you put them together, it's the development of a new vaccine. And we have to do all the clinical studies, we have to generate the clinical data on immunogenicity, on safety, as it would be a completely new vaccine, because you never know how these antigens behave when you put them together. So regarding Hexatzima, it took us about 10 years from the start of the project to license this year. And the production time is also long for such a combination vaccine. It takes up to 33 months to produce one batch. So especially working then with governments, when they want to have big quantities of vaccines, it really needs to have some cooperation. We cannot provide the vaccine from one day to the other. We need to plan with our customers to have the vaccine ready in time. Let's look at the vaccine antigens now. So we have on the one side the established antigens that we use since many years in Sanofi Pasteur: diphtheria, tetanus, acellular pertussis, IPV, the PRPT, the HIP antigens, used either standalone and more as combination vaccines. And you know very well the pentaxime, for instance, where the 5 antigens are in one. And we added our new hepatitis B antigens to this new Hexatzima. A few words about the manufacturing process. So this is a very busy slide, but let me say a few words about it. So we need to produce separately all these different antigens. We have to produce the diphtheria toxoid, the tetanus toxoid, the 2 pertussis antigens that are in our vaccine, so pertussis toxin and filamentous hemagglutinin. And also here, these are 2 different production processes. Pertussis toxin is detoxified with glutaraldehyde. Our filamentous hemagglutinin is not detoxified, it's in its native form, and we use chromatography column purification. Then we have to produce the 3 polio antigens on vero cells, the HIP antigen, PRPT, so the PRP that needs to be conjugated to a protein, tetanus protein in our case, to be immunogenic in the young infants, and finally the hepatitis B antigen. So it protects against 6 diseases, but in fact it's more than 6 antigens, it's 10 antigens if you count them, 3 polio antigens, 2 pertussis, and this tetanus protein is another one. And after blending, there are still more than 30 tests to be done to validate the product. And altogether in the production process, it's around 150 tests for validation, quality tests. So as mentioned, it takes up to 33 months to produce one batch. So the biggest part of the time is the production of the different antigens that we have just seen. And just to mention, there are also dedicated buildings for each of the antigens. It's only diphtheria and HIP that are produced in the same building, but at a different time. And for all the antigens, there are separate buildings dedicated just for the production of the antigens against one specific disease. And then it takes about 9 months for pharmaceutical form, packaging, all the quality issues. I'd like to speak a little bit about the hepatitis B antigen. This is the new antigen in this vaccine. We produce it with the Hansenula polymorpha yeast expression system. So this is a yeast expression system that has been initially developed by a German company, Rhein Biotech, and Sanofi Pasteur acquired one part of this company. And we developed this yeast expression system further to make it suitable for large-scale production process. And the advantage is that it gives a consistent high quality and a very reliable supply. It is for sure compliant with European Pharmacopoeia, with WHO technical requirements, and we produce it exclusively for the use in Hexazyma. So we focus all our production capacity for hepatitis B antigen to put it in the combination vaccine. It has also experienced this yeast expression system for producing other proteins like insulin or interferon alpha. So this is an animated slide. I will not speak a lot about it. It shows the production process of the HBs antigen, the fermentation, then you see how it goes on. And also here it takes 4 months, 11 quality control tests to release one batch of HBs antigen. So the first activities we did, first studies we did, is to look at the immunogenicity for the hepatitis B antigen. So we tested it in 2 groups, in adolescents, that's at the left side, adolescents 10-16 years, and then in the group 16-45 years, different amount of antigen, 10 microgram, 20 microgram, and we compare it to control hepatitis B vaccine, in this case was Engerix B, so a very widely used hepatitis B vaccine. And what came out is a very consistent and high immune response. And based on these results, Sanofi Pasteur decided to use the 10 microgram formulation to include it in Hexazyma. And all the further studies done were already done with the combination and not anymore with the standalone hepatitis B antigen. On this slide, you see the composition of Hexazyma. So it's a 0.5 milliliter vaccine. I will not read all this, I'll let you have a look at it, and you know it from the pentaxime. It's very similar. Regarding adjuvant, it's adsorbed to aluminum hydroxide, and it's preservative-free. So it's pentaxime and 10 microgram of the new hepatitis B antigen. If you look very carefully, you will see one difference that's here for the PRPT, for the HIP antigen. This is 12 microgram. And in most of the HIP vaccines, in most of the combination vaccines, it's 10 microgram. Here it's 12 microgram because this is a fully liquid vaccine. And the big challenge is to put the HIP in a liquid form and to keep it stable in the liquid form. And until now, no one else succeeded to do it. And our production people took the challenge to put it in the fully liquid form, and they succeeded. From a technical point of view, it's a very difficult point. But it was shown to be stable and then to produce the same immunogenicity results. A big advantage of Hexazyma is that it's fully liquid, which means fewer administration steps. And here on this slide, we see a comparison between the fully liquid vaccine and the vaccine that needs reconstitution. I will not read all the different steps. But if we count it, it's 11 administration steps less using the fully liquid vaccine, which means quite some time saving and quite some additional comfort, less stress for the parents, the baby, because everything goes quicker. And also no danger to do anything wrong with the reconstitution. Now I'd like to give you a brief summary about the clinical development. So we had a comprehensive development plan. We did 13 large-scale studies, including more than 5,000 subjects. We did immunogenicity and safety evaluation. We evaluate the primary vaccination schedule, first year of life, and the booster in the second year of life. We did 1 large-scale safety study, lot-to-lot consistency studies, co-administration with other pediatric vaccines, and also looking at long-term antibody persistence. So here are the vaccination schedules that we studied. We studied the 6, 10, 14 weeks WHO EPI schedule, 2, 3, 4 month schedule, 2, 4, 6 month schedule, with and without hepatitis B at birth, and then the booster dose. I know that the 6, 10, 14 week schedules in Europe has no importance, but I will speak a few times about it because from an immunological point of view, it's the most challenging schedule. It's the most challenging to get a good immune answer starting at 6 weeks of age and having these short distances between the vaccinations. That's why I will speak also a little bit about it. Also, I know for sure it's not used in our area. The control vaccines that were used in the studies were either acellular pertussis combination vaccines, pentavalent, hexavalent, or also wholesale pertussis combination vaccines. And the concomitant vaccines used in the studies were pneumococcal conjugate, rotavirus, MMR, and varicella. And I will show you some of the results in the following slides. So here we see immunogenicity data after primary vaccination for each of the antigens. So we see the seroprotection, seroconversion rates for each of the antigens in different schedules, 6 and 14 weeks, 2, 3, 4 months, and 2, 4, 6 months. And the message here is that independent of the schedule used, the antibody answers are very good. And then regarding comparison. So here we compare it to Pentaxime, to the pentavalent vaccine. We looked at seroprotection, seroconversion rates after the third dose, and the schedule here was 2 for 6 months of age. And we see it for all the different antigens. On the left side, this dark color is always with the hexatzima, and the shaded color is with the Pentaxime and standalone hepatitis B vaccine. So the message is that the immune response to all the antigens is high in this comparison, similar to that of Pentaxime, co-administered with a standalone hepatitis B vaccine. We compared it also to the other licensed hexavalent vaccines, so to Infanrix Hexa, in the schedule 2 for 6 months of age. So the graphs are the same, seroprotection, seroconversion rates for the two groups. And what comes out is that, again, the immune response to all the antigens is high, is similar to that of the other licensed hexavalent vaccine. So very good immunogenicity data. Let's focus a little bit more on hepatitis B. And here we look at this challenging schedule, the 6, 10, 14-week schedule, which, as I mentioned, is really from an immunological point of view the most challenging. One group of the infants received hexatzima, 6, 10, 14 weeks of age. One group received the standalone hepatitis B vaccine, and you see that seroprotection, so defined as anti-HBs antigen at least 10 milli-international units per milliliter, is very similar. If we add the hepatitis B vaccine at birth, the seroprotection rates are a little bit higher, the GMTs are much higher, but even without the hepatitis B at birth, the immune answer in this challenging schedule is very good. And that's the reason why hexatzima is the only hexavalent that is licensed for use with this EPI schedule in the absence of hepatitis B vaccination at birth. We stay with hepatitis B, we go back to the more usual schedule, 2 for 6 months of age, and we compare it to Infanrix Hexa without hepatitis B vaccine at birth, with hepatitis B vaccine at birth. The green group is the hexatzima, and we see that also here the immune answer is very good. We get consistently high immune answers to the hepatitis B antigen in the different schedules with or without hepatitis B at birth. So we have seen that it's highly immunogenic against hepatitis B under various vaccination schedules, also in the absence of hepatitis B vaccination at birth, even under this challenging, immunologically challenging EPI schedule, and we are the only one to have this license without hepatitis B vaccine at birth. Regarding the booster responses, so the vaccination in the second year of life, I will not show you the graphs and the bars now because I know it's tiring. I will give you a brief summary. So it has been given as a booster in children either primed with hexatzima in the different schedules that we have seen, or in children primed with Pentaxime and standalone hepatitis B vaccine, or in children primed with Infanrix Hexa. And the result of all these studies is that regardless of the primary series vaccine, the hexatzima booster results in a good, a very strong anamnestic response to all antigens. So these data have been published now. You can see the publication. As mentioned, I will not show you graphs and bars now because it always looks the same. Let's focus on safety. Here in this study, we compared hexatzima to Pentaxime given with standalone hepatitis B vaccine. And we look at local reactions, pain, redness, swelling. So the green group is the hexatzima group, and the other group received hepatitis B and pentaxim. So what comes out clearly here that, I mean, if you add these two together, it's clearly more reactogenic than just giving one vaccine. So this shows that the combination vaccine induces the injection site reactions that are associated with multiple injections. Here I show you some data where hexatzima was compared to Infanrix hexa. It's a quite busy slide. So on the top bar, we see the percentage of subjects with any solicited injection site and also systemic reactions after any dose. So this is clinical study setting. And you ask the parents for every reactions to notify it. So the percentages are very high. So it goes for irritability over 80%, pain over 80%, which is the same for both vaccines. This is not the reality. This is not the clinical reality that you face. The clinical reality is rather this, the grade 3 reactions, when there's really a problem, when there's a more strong adverse event. And here we see that it's very well tolerated and that these more severe adverse reactions are very rare. So we have shown in all these studies that in the different schedules that hexatzima is safe and well tolerated, also for the booster dose. For the booster dose, regardless of the primary schedule and regardless of the primary vaccine given, there is no difference. It's clearly better tolerated than wholesale-based pertussis combination vaccine. If we look at the most frequently reported reactions, it's not surprising. It's injection site pain, it's irritability, crying, and injection site erythema, as we would expect it for such a pediatric vaccine. Hexatzima has a comparable safety profile to other licensed acellular pertussis-based combination vaccine, which are very well tolerated, and it can be safely administered concomitantly with other pediatric vaccines. So to summarize all these clinical trials, these 13 large-scale studies enrolling more than 5,000 infants showed a similar and a high immune response to all the antigens, post-primary, post-booster. And we have already seen all the comparisons. Hexatzima induces also very good booster immune response, regardless of the primary series vaccine. And as stated in our SPC, it can be co-administered with pneumococcal conjugate, with rotavirus, and with MMR vaccines. So we can provide you now an innovative hexavalent vaccines. All these antigens are fully manufactured by Sanofi Pasteur, which means for us that we really have the control over the production process of all these antigens, and we can assure the high quality. We developed this recombinant hepatitis B surface antigen specifically to be included in the hexavalent vaccine. It has for sure the improved convenience and compliance via the 6-in-1, reducing the number of injections, reducing the number of medical visits. It is a fully liquid, ready-to-use vaccine. No reconstitution required, as we have seen. It means many administration steps less than reconstituting a vaccine, so administration time is reduced, less stress for the parents, less stress for the baby. And it ensures also the right dose of each antigen every time. I think sometimes mistakes can happen in reconstitution here. There is no risk. And as mentioned in the introduction, it was registered in springtime in Europe via the centralized procedure. We register it all over the world now. It's already used in some countries. We started to use it in Germany. Germany was the first country really using it on large scale since this summer. And with a very good success, doctors are very satisfied. Also in my area, mainly the Central European countries, we already won some tenders, for instance in Romania. So we spread it out now over many countries. Thank you for your attention. Efaristo.